this is an example from our curation of data sets, the metadata curation for data sets. And these data sets, they are uploaded typically by scientists to sharing sites like ArrayExpress or Gene Expression Omnibus, which we are very grateful because that gives information to all of us that we can use to further science. However, for the scientist that is required to upload these to get his paper published, it's often just kind of an additional hurdle to jump over to publish them to this site. And you can see this by the data submissions. So in our experience, more than a third of all of these submissions that get uploaded have some error, some typos in the text. You know, the cell line is misnamed, there's typos in the treatment and stuff like that. Now, this is something you can easily fix with human curators because you can tell from the context how that cell line should be named. We also do find about 5% of the uploaded data sets contain errors that are really errors. There are mislabeled treatments, mislabeled sample groups. And in closing, I'll just show you one of these to give you an idea what you're dealing with. So this is a shall be unnamed data set from Gene Expression Omnibus, where an experiment was done between two genetic subgroups of, I think it was B-cell lymphoma or something, that were termed B other or non-B other. And what you see in this table is these are patient IDs in this column down here. In these two columns, you see what the age was that was reported for the patient, and here which group it belonged to. And the group is important because that was the core of the entire study. You know, is there a difference between these two groups? Now, the thing that you can see from this study here is that from all the patients on this list, there's only one patient where the same age was reported in the uploaded data set and in the paper. All the others had different ages. And even worse, you know, half of them were grouped in different groups in the uploaded data set and in the paper. So this is something you cannot resolve without contacting the authors, which we do, right? You need to get in contact with them and find out what happened there if you really want to know what was the actual group that this patient was in. Also, if you're only looking at Gene Expression Omnibus and not even checking and reading the publication, then you'll never find out that this data may be messed up.
Curation.
Sample to insight.